So hi everyone, I'm thrilled to be here to share some of the research that I've been doing. So I'm a postdoc researcher at the Northeast Climate Adaptation Science Center, which is based at the University of Massachusetts, obviously in Massachusetts. And the title of my talk today is Invasive Plant Abundance Habitat. So identifying high-risk range-shifting taxa using EBMAPs. And so the second half of this talk, we're going to be going through a relatively new online tool that you can use to identify potentially high-risk range-shifting taxa. And to start off with, I want to give a little bit of background about how we developed this tool, so where the data and the outputs came from. Be great if I could advance. There we go. So I'm assuming that most of the people who are on the call today are relatively familiar with how challenging it can be to both monitor and manage for invasive species. And now in this sort of realm of climate change, we're having to not only manage for current invasive impacts and species, but also those impacts and new range-shifting species as a result of climate change. And we know that climate change can impact all stages of the invasion process. So just to give you a couple of examples, so through melting of sea ice, for example, we can see shifts in the pathways that invasive species can take towards being introduced into new areas. We also see climate impacting establishment of invasive species, so shifts to disturbance regimes, and also changes in climate suitability can help invasive species establish in places that they currently can't establish or establish more rapidly. And that in turn also impacts the spread of present invasive species, so invasive species that are already established in a region. So, for example, for invasive plants, spread can be facilitated by longer growing seasons. And we also see changes in the competitive landscape as a result of climate change. And these changes in competitive landscape can then in turn affect the impacts that invasive species might have on native taxa or communities. So we might see shifts in invasive species abundance, which in turn lead to shifts in whole community dynamics. And so one of the most common ways that we often think or study the impacts of climate change is on the geographic distribution of invasive species. And one of the main concerns is that species might need to track suitable climatic conditions across the landscape, in which case we're going to be seeing invasive species moving much more rapidly than we might have seen a couple of decades ago. And despite all those negative impacts of climate change that I showed you on the previous slide, there is some optimism and hope, and that's that for a lot of these range-shifting species, they haven't had the opportunity to either move or establish in these new regions yet. And so if we're able to identify those species before they're able to establish large populations, then we can proactively manage them. So sort of think about it as being management in very early stages of this invasion process, focusing on both prevention and eradication. And so over the past several decades, there's been a ton of work invested in ways that we could identify what species are likely to be shifting their ranges and where they're likely to be shifting to. And one of those studies was done by colleagues of mine, Jenica Allen and Bethany Bradley. So in 2016, they ran a variety of species distribution models using occurrence data of invasive plant species that are already established in the United States. And here I'm showing you one of their key figures. And so this is the number of new invasive plant species that are likely to be able to establish in regions in the northeastern United States, given future climatic changes, so around 2,100. And so the darker the red color in this figure, the higher the number of invasive plant taxa that could establish populations there. And they use these results to create a listing tool, which I'm going to be going through a little bit later. And so this is a really great first step towards identifying species that are likely to pose the greatest risk. But as you can see here in this figure, the scale goes up to 100, and 100 invasive species on top of ones that we're already trying to manage is quite a lot. And so this kind of led to this question of, okay, that's great, but which species should we prioritize now and which can we maybe not be quite as concerned about in terms of proactive monitoring and management? And so there's a couple of different ways that we could narrow down a list of potentially range-shifting taxa. And so one of those is to not just focus on where a species could occur on the landscape, but where they could reach some level of high abundance. And this is because often where species are able to reach high abundance is a much smaller subset of the species range compared to where they could just occur. So here I'm showing you an example of what that could look like. So the yellow regions, these are two different invasive plants. And so the yellow regions are showing areas where species distribution models predict these species could simply occur on the landscape. So think of this as like a presence absence estimate. Whereas the dark blue areas are where these populations could not just establish, but also reach some high level of abundance. And so you can see that they're a much smaller subset of this full range. Another reason that we might be interested in looking at high abundant populations or figuring out where species could reach high abundance is because often high abundance is correlated with higher impact. So if you think about if we've just got a single organism or population on the landscape, that's much easier to both manage and monitor for than it is trying to manage a very large and broad sprawling population of high density. So that's great. We can focus on abundant populations, but this begs the question, okay, which species are likely to become abundant and where are they likely to become abundant with climate change? And so this is the main question that's driven a lot of the research that I've been doing in the last couple of years. And so to answer this question, my work is focusing on invasive plants. And so for the rest of the talk, all the results I'm presenting are based on invasive plant species that are already established in the United States. And so we combed online databases, 18 to be specific, looking for abundance data on any invasive plant currently established in the US. And when I say abundance data, I'm referring to percent cover metrics. So there's a couple of different ways you could measure abundance. And so we're using percent cover. And we're classified a species as abundant if they could reach at least 5% cover of the landscape. And so after a variety of pre-processing and checking and cleaning of that data, we ended up with a candidate list of around 144 invasive plant species. And we focused these species for our species distribution models. And to start off with, we were really interested in just truncating that list to species that are likely to have an East Coast distribution. But we have subsequently rerun the analyses for a different set of West Coast taxa, which I'll give you a sneak peek on later on. And so with colleagues at USGS out in Colorado, we ran a variety of species distribution models. And so for each of these 144 taxa, we ran 5 different species distribution model algorithms using a suite of current climatic conditions that involved a variety of temperature and moisture variables. And then we also ran models based on a future climate scenario of around 2 degrees Celsius of climate warming, which we're expected to reach around 2100. And I'm happy to dig more into these methods in the Q&A, but for now, I just want to keep them short and sweet so that we can move on to the results. And so here, I'm just going to jump right into the results and show you where the current hotspots of abundance habitat is. And by abundance habitat, I mean areas that are climatically suitable to support abundant populations or populations that can reach about 5% cover at least. And so again, to start off with, we're focused on the East Coast of the United States. And so here, I'm showing you that East Coast region, and the darker the red color of this map, the greater the number of species that are likely to be able to establish high abundant populations in these areas. And what we see is that based on current climatic conditions, there are sort of 3 general regions that serve as hotspots where a large number of invasive taxa can reach high abundance. And those are the northeast coast of Florida and Georgia, so down here, the Great Lakes region and the Mid-Atlantic region. So you can kind of see this big band of red sort of in the north of the United States. And so this is sort of the status quo of what things look like at the moment. And the second part of our question was how these locations of abundant suitable habitat, or these hotspot areas, are likely to shift with 2 degrees of warming. And so here I'm showing you what the output map looks like for that. And so I guess I should also clarify the way that these maps were produced is that for those 144 taxa that we have models for, we essentially took all of the model outputs and predictions and stacked them on top of each other to produce this one big composite map. And so you can see on the right-hand side for the future hotspots, so under 2 degrees Celsius of warming, We see overall the regions of climatically suitable habitat for abundant populations of these invasive species are likely to shift northwards. So mostly towards the northeast, followed by the northwest, as these plants are tracking climatic conditions that are suitable for their populations. And what's interesting is we actually see an overall average range shift of around 112 kilometers for these invasive plants. As I mentioned, we also repeated all of the methods that I described earlier for a suite of West Coast species, the species that are currently present on the western side of the United States. These are really preliminary data. So here in this figure, the darker the red area, the higher the number of species that could have abundant populations in these regions. And we also see sort of 3 general hotspot areas based on current climatic conditions. So particularly along the Californian coast, we see a large swath of the central Great Plains, and then also the Columbian Plateau up north that seem to support quite a high number of invasive plants that could reach high abundance. Again, we could ask the same question: how are these hotspot areas likely to change with 2 degrees Celsius of climate warming? And we see that much like the East Coast, most species are shifting on average towards the northeast. But what's really interesting is we do see quite a large number of taxa shifting their range southeast. My guess is that a lot of that is being concentrated in areas such as this Californian coastline, although I haven't had much of a chance to really dig into what species are shifting where and if we can explain why. But what we do see is that compared to the East Coast, the average range shift of species out west is much larger. So we're expecting to see abundance ranges shifting on average 317 kilometers, so about twice the distance that we see out east. And as you can imagine, when we're modeling a whole bunch of different species, there's quite a wide range of responses that we can expect to see in individual taxa. And so for those 144 invasive species that we ran models for our East Coast data set, we can also tease apart and plot out how individual species themselves are likely to be moving. And so here I'm showing you examples of just what 2 of those potential responses to climate change could look like. And so here on the left, we have wine raspberry as an example of a species that is predicted to expand its overall abundance range, or the amount of habitat that's suitable for abundant populations. And so here we see quite a large area of the species range that is predicted to become unsuitable with climate change. And so that's shown in the blue color here. So these are areas that are currently suitable, but are likely to become unsuitable with climate change. We see a large area that's predicted to increase in suitability in the future. So the red areas currently are not suitable, but are predicted to become suitable for abundant populations with climate change. And then also a large area of their current range, shown in purple, is likely to still be suitable for these abundant populations in the future. And so it's these sort of red and purple areas where management is likely to need to continue or ramp up in the future. We kind of see the reverse trend for alligator weed shown here on the right. So although it is showing this overall northeast shift, and you can see that there is a band of new habitat that's likely to become suitable under future climatic conditions that maybe isn't suitable now, again shown in red. A large area of its current range is predicted to become unsuitable with climate change. So that's shown here in the blue. And it's these areas in blue that could offer an opportunity for restoration and for shifts in how we're allocating our management resources if conditions are not conducive for the species to survive. So as I mentioned, we've got output like this for all of those species, but we've also created an alternative way to access the information from these species distribution models. And we've done that in the form of state lists. And so I'm going to use Massachusetts as a case study because that's where I'm based. So out of those 144 taxa that we modeled for the East Coast, 108 of those are predicted to have climatically suitable habitat for abundant populations in the future. 45 species are predicted to actually increase in their abundance habitat in the future. So if they currently have habitat, it's likely to expand, or there's 7 species which currently don't have habitat, but are likely to have suitable habitat under 2 degrees of climate change. And I'm showing you those species, or a picture of those species here on the slides. And so again, these are species that don't currently or are unlikely to be able to sustain abundant populations at present, but are likely to be able to in the future. And so it's these ones in particular that monitoring should be focused on so that we can eradicate them before they're really able to establish. And so I'm going to switch gears for the last little bit of my talk and share with you one of the ways that you can access this information that's already available to you online, and that's through the EDMAPS website, and specifically through the invasive range expanders listing tool. And so if you're unfamiliar with EDMAPS or this listing tool, I'm just going to take a few moments to walk you through how to use it and how to access the information that I've shared with you. So this invasive range expanders listing tool was developed as a result of the output data that Jenica Allen had created through her species distribution models. So one of the very first maps that I showed you in this presentation. And so as a reminder, Jenica's models were just based on occurrence data. And so that was what this range expanders listing tool was initially designed to do. So if you go to the EDMAPS website, and you can click on this tools and training button at the top, and it will take you to a drop-down list, and you can see here I've circled this range expanders listing tool. And when you click on the tool, it takes you to a page that looks like this. And so this, you can select the state that you're focusing your management or are mostly working in. If you're only working in a particular county, you can select that from the list, or you can look at all counties. Or you can also select species within a particular radius. So maybe you're also interested in species that might be shared by a neighboring state, for example. And then you can also select the number of models that predict suitable habitat for that species. So for Jenica's work, it was between 1 and 13 models. So the higher the number of models, the greater the certainty or agreement between models that that area is likely to be suitable in the future. And what you'll see as you're toggling between all these different drop-down menus is this map dynamically update to show the region where data is being pulled from, and also a list of species. So it basically creates a watch list and updates that watch list dynamically for you as you're toggling around. And in addition to that, you can also click on individual taxa or search for individual taxa. So maybe you're really interested in, say, managing for Japanese knotweed. And so you can open up a page, an information page specific to that species, and you can see there's a variety of information points that you can look through at the top. And if you click on the counties button for Japanese knotweed, it will open up this menu underneath. And it can take you to this tab here of future certainty or future range. I'm going to talk about future certainty. And so here it will create a map for you that you can zoom in and out on, and it will show you in terms of model agreement, so how many of the 13 models agree that that area is likely suitable for that species to occur. And you'll notice that for the species that I've been modeling for my abundance work, that there's this new tab here called Future Abundance Habitat. So what I'm showing you on the screen at the moment is just based on occurrence data, but now we've also added the abundance data so that we can try to narrow down this range to just identify the counties where Japanese knotweed, for example, could not only occur, but also reach high abundance with climate change. And so if we click on this Future Abundance Habitat tab, it'll take you to a very similar looking map. This time the scale's out of 15, but same deal, the larger the number, the more models agree that that area is likely to be suitable. And so you can toggle backwards and forwards between these two to compare where the species is likely to just occur on the landscape versus regions where it's likely to also reach high abundance. And it's our hope that this is going to be useful for narrowing down that list of potential range shifting species. And at the moment, this data is available for you to use and access for 215 species, so both the East Coast and the West Coast taxa. And so here are a variety of links that you might find useful if you're interested in using this tool. I'm also a member of the Northeast Risk Management Network, and if you go to the Northeast Risk website, there is a link to a YouTube video which has an online tutorial that goes into a bit more detail about how to navigate around some of the tools of this range expanders listing tool. So if you're interested, you can pull through the website and find that, or send me an email. And with that, I'd just like to thank all my co-authors and funding sources and all the people that have made this project, both the research side and getting it uploaded onto EdMaps, possible. And with that, I'll take any questions, and you can also reach me at the email address on the slide.
Thank you, Annette. Unfortunately, we don't have time for questions, but it looks like you do have a couple in the chat. So I encourage you to answer those within the chat, but make sure you post that, kind of copy the question and post to everyone so that they are able to see those questions as well. So
Sure.
Thank you very much, Annette.
Thanks, everyone.
Okay, our next speaker is Claire Tortorelli. She is from the University of California, Davis. So Claire, if you're on, go ahead and share your video and your screen, and I'll leave you to it.
Great, thanks, Jen.
Yep.
Okay, does that look good? Can you hear me okay?
That looks great, and you sound good.
Great. Yeah, thanks. I'm really excited to be here today and to hear all about everyone's exciting work and to share some of my PhD work at Oregon State on Ventonata dubia, some more recently introduced annual grass, its invasion throughout the Blue Mountain ecoregions, and its relationship with fire. So before I really dive into this work, I just want to express that this was a huge collaborative project. Wouldn't have been able to do any of this work without support from Becky Kearns, kind of the Ventonata project, one of the project PIs at the USFS Northwest Research Station, along with many others at Oregon State, the Rocky Mountain Research Station, work with Pyrologics for some of our fire modeling, many field techs, and work with Ellie Dean at the BLM. So many of you are probably really familiar with this idea of the grass fire cycle, or this positive feedback loop between grass invasion and fire in some of our maybe less frequent or less fire prone ecosystems. So one of the big examples of this is the cheatgrass invasion in the Great Basin, where you get areas like this, where we're seeing this photo on the left of big sagebrush with lots of bare ground interspaces, kind of our perennial bunch grasses. And then pre-invasion, and then once cheatgrass comes in, it really fills in those interspaces, increasing fuel loads and continuity in this previously rather sparsely fueled, sparsely vegetated ecosystem. And that can facilitate larger fires and more extensive fires throughout the region. Because these annual grasses typically recover very quickly after fire, this can also lead to more frequent fires in this kind of positive feedback cycle of fire, invasion, and more fire. And like I mentioned, these grass fire cycles are not a new phenomena for the world, and especially for the Western United States. We've known about kind of cheatgrass invasion in Medusa Head in the Great Basin for a long time. We also see similar grass fire cycles in the Mediterranean regions of California and in the Southwest, as well as some cheatgrass in the Columbian Basin. But there's some regions that we don't really think about these grass fire cycles, predominantly our forested ecoregions. And for the rest of this talk, I'll be focusing on kind of this jelly bean shape cut out in northeastern Oregon called the Blue Mountains ecoregion. So this is a really interesting kind of case study for an invasive species, especially because this is kind of a forested ecoregion, but if you look closely at the map, you'll see it's not truly just homogeneous forest, right? We have these, it's really more of a forest mosaic with many patches of non-forest. This is mostly kind of low sagebrush, we'll call these kind of scab lands, these non-forested areas, surrounded by, embedded in this kind of larger forested matrix. So interesting about this area, and also really problematic is this more recently introduced annual grass ventinata has heavily invaded many of these non-forest scabland patches. And this is causing a lot of concern because these areas were historically really resistant to other annual grasses, including Medusa head and cheatgrass, even though they kind of exist in this ecoregion in the big sage patches around. These scablands have been really relatively resistant. Ground ventinata, some of you are probably really familiar, some of you may have not heard of it. Some of the common names are the genus name ventinata, wiregrass, North Africa grass. It's a fall germinating annual, like cheatgrass and Medusa head has many similar characteristics to those other common invaders. Relatively short-lived seed bank. It has really shallow roots, like less than 2 centimeters in many cases. So not a lot of infrastructure there underground. It senesces very early in the summer, much earlier than our native perennial grasses, and it can grow in these near monocultures, especially in these scablands. Ventinata was introduced in the early 1950s, kind of this corner between Washington, Oregon, and Idaho. And its native range is across much of Europe or kind of Eurasia region. And it's actually a rare species in its native range. It's listed on their rare species list, on their red list. But it just has kind of taken off, spreading it over 1 million hectares per year here in the Northwest, and really down into Wyoming and a little bit into California, as well as some of our eastern states. So ventinata, like I mentioned, has been really heavily invading these historically non-forest invasion resistant scabland patches. And that could be very concerning for the biodiversity in these patches that didn't evolve with fire really at all. These are very sparsely vegetated areas. They hold a lot of endemic species, and they historically really just didn't burn. If they did, it was very patchy, very isolated burns. And now we're seeing these extensive burns across these scablands, likely fueled by all of this increase in fuels by ventinata. And so this could be really problematic for fire management. And here's a little video I'm going to share from a friend, Will Downing. He was a smoke jumper for the Forest Service. And you see here, he's accessing a fire that's burning in one of these forested regions right next to this scabland patch. And in this case, the scabland is not invaded. So there's really not any fuels in this scabland to carry that fire across or into the scabland or into the surrounding forest. So it's a relatively isolated fire, also with a lot of access area to kind of get this fire under control before it has a chance to spread. But you can imagine if all this bare ground in these scablands was full of that very flammable annual grass ventinata, that the story could be a little bit different. We might see much more fire spread across this greater mosaic. So it leads us to kind of our first questions, or to really about how can we manage this species? It's rapidly expanding. We don't really know anything about it because it's rare in its native range. So in order to prioritize areas for prevention, for monitoring, and for treatment, we need to first know what are the areas of highest risk, and how does ventinata differ from other invasive grasses? And so we know some of this just from our observations, but some of my PhD work was to go ahead and investigate this a little bit further by sampling fires across eastern Oregon, across the Blue Mountain ecoregion, where we looked at vegetation and site characteristics and tried to kind of characterize ventinata's niche in relation to other annual grasses, cheatgrass and Medusa head. And so I'm going to show this plot here. We'll get a little familiar with it. What you really need to know is that the dots on the plot represent our dots in this plot represent our sample plots. And the location in this space is how similar they are in species composition. So I've colored and shaped these points by their plant associations. And so it kind of makes sense here. We've got a lot of green dots together. Those are going to be more similar in species composition in our open conifer forests, whereas our scablands kind of fall out down here, and they're also tend to be more similar in their species composition. And then I've gone ahead and kind of superimposed our environmental variables on these axes. So in axis 1, we have increasing kind of rockiness to our soils, and axis 2, kind of increasing in canopy cover and kind of decreasing in temperature. And then what I've done is made almost made like a heat map of Ventonata in this kind of, we call it species space. So you can see which kind of vegetation types and which environmental variables Ventonata is most strongly associated with. And what we found is that, like we expected, Ventonata was really highly associated with these scabland patches. But we also saw a lot of Ventonata in our open forest, which was a little bit surprising, up to even about 50 to 60% canopy cover. You're still seeing pretty high abundances of Ventonata in these unburned forested areas. So then we did the same thing with Medusa head and cheatgrass, and we essentially just kind of drew polygons around the reddest regions of these heat maps so we could compare their distributions. And we found that Ventonata, again, is in those scablands, open forests. Medusa head also likes the scablands, but at lower elevations and tend to be really, really clayey soils, rocky soils. And our cheatgrass tended to be on sandier soils, mostly kind of in our big sagebrush areas. But where these three species overlapped was in our severely burned forest, especially if it was close to one of those scablands. So I'm not showing here our relationships to fire, but we did the same thing looking at fire variables. We found that while these species tend to be pretty distinct in these unburned sites, they really overlap in those burned forested regions. So this is kind of extra concerning, right? We're not just getting Ventonata in areas that are already heavily invaded by cheatgrass and Medusa head, where it might be causing additional problems, but kind of already impacting areas that are heavily impacted. We're seeing Ventonata invading this kind of completely novel niche. So really expanding that invasive grass footprint across areas that historically were pretty invasion resistant. Let's talk a little bit more about Ventonata and fire now. So we saw that video of these scablands kind of acting as these natural fire breaks. So what happens when those scablands are heavily invaded by Ventonata? How does that impact landscape scale fire behavior? So again, just a reminder, here's a picture of uninvaded scabland on the left, invaded scabland on the right. And we want to know, what does this mean for fire behavior in these scablands, but beyond that at the landscape scale? So you probably don't need science to tell you that there's more fuel in this scabland on the right, but we went ahead and measured it and found that Ventonata increased fine fuel biomass by 50 times in these invaded systems compared to the uninvaded systems. So huge increases in invasive fuels, and also seeing, or just in fine fuels in general, and seeing much higher continuity, so fewer bare spaces between these fuels. So what does this mean for landscape scale fire? We simulated fire across the entire ecoregion using FSIM, a large fire simulator. It takes into account things like topography, historic fire, weather, fuels, and ignitions, and outputs maps of burn probability, as well as kind of burn parameters for all of the simulated fires and flame lengths. So we ran FSIM twice on the entire Blue Mountain ecoregion, once with our kind of uninvaded landscape, as is with low fuels in the scablands, and once where we added fuels, where we have mapped the Ventonata invasion. And so this was mapping the Ventonata invasion throughout the ecoregion, was a project done by Ty Natupsky, and this is one of his PhD chapters to develop this species distribution model for Ventonata, and actually kind of more of a map. So this is like where it is today. And we increased fuels in the scablands where Ventonata was invading. And then we compared those fire outputs. But what we were most interested in is really, how is fire transitioning between scablands and the other areas? We kind of expect there to be more fire in the scablands because we've increased fuels in the scablands. But what else is going on at the landscape scale? So we measured all the fire transitions in the area burned from fires ignited in our vegetation types here on the y-axis, and we looked at how much of vegetation types, how much area of these vegetation types on the x-axis those fires burned into. So for example, fires, we saw a big increase in the amount of area burned in closed and open canopy forests from fires ignited in the scablands. So we saw a 15% increase, a 30% overall increase in the amount of area forest burned because of fires ignited in scablands with invasion than without. And this went both ways too. We saw 17 and 20% increase in the amount of scablands burned from fires that were ignited in our open and closed canopy forests. So we know there is more fire in the forest because of invasion, but what is the characteristics of that fire? Is this kind of lower severity, higher? What's the probability that this fire in this forest is going to be kind of high severity or require a shift in our management practices because of invasion? And so to look at that, we use that same simulated fire behavior, and we ask kind of what happens when you're a forested pixel and the amount of invasion in your surrounding neighborhood changes. So for example, a forest that's surrounded by 50% of its neighborhood is invaded is 9% more likely to burn with flame lengths greater than 4 feet than if none of the surrounding neighborhood was invaded. So that might seem like a small amount, but an increase in those flame lengths can also shift our management practices. So instead of being able to control this fire now with hand lines, we're more likely to need large machinery. And the same can be true in the scablands, where now, maybe before we didn't have any fire, now we have more fire. So we're just kind of pulling more resources to manage fire because of this invasive grass. So to wrap it all up, we found that increased forest fire frequency may promote this forest structure, but that post-fire invasion might impede forest recovery. So this is kind of speculating maybe future work to look at how invasion into these forests that have lost their canopy are going to recover now that the understory is heavily invaded. Hand-lined may be less affected in invaded areas. We might need to bring in machinery, and machinery can exacerbate invasion, so it may be another one of these positive feedback cycles. We might need to rethink fuel treatments in our scablands and around our scablands if these are no longer acting as these kind of natural fire breaks. And these impacts may just continue to increase as we predict the invasion will continue to spread. So this is some of our work. We have a lot more work in progress, and also that's been completed in other aspects we just didn't have time to talk about today. We've also looked at kind of the future distribution of Ventonata across the entire West, not just Oregon, as well as we're doing future fire models for the Blue Mountain ecoregion. And you can see kind of all of our work, our publications at this website. Feel free to email me with questions. And yeah, if I have time for questions, I could take some now. Thanks.
Yeah, it looks like we have a couple minutes. It's like we have one question asking how the impacts from the Ventonata compare to cheatgrass, or about how you think climate change might be affecting these dynamics.
Yeah, that's a great question. I don't think we have a great comparison for like total impact of Ventonata versus cheatgrass, because they're really, in many areas, impacting very different ecosystems. I mean, they seem similar because they're both sagebrush, but Ventonata seems to invade more of these like scabland, really rigid and low sage sites, whereas cheatgrass is invading more large sage. I'd imagine that their impacts are going to be very similar, though, right? We are seeing it's likely we're going to see more competition with those native species, especially probably reseeding sagebrush after fire when you already have a lot of Ventonata present, although we really don't have any research on that, whereas there's a lot more research that's been done on cheatgrass. We may also see kind of loss of that soil structure because of the really shallow root system, maybe even more so than cheatgrass, because Ventonata has much shallower roots than cheatgrass. And I expect these problems to continue to exacerbate with climate change, especially the relationships with fire, as we continue to see hotter and drier fire seasons in the Pacific Northwest. Our fire seasons continue to get longer. Our grasses are probably going to dry out sooner. So I imagine to see these problems just continue to persist as our fire problems keep getting worse and our invasion problems keep getting worse. But yeah, I think that I didn't really mention control because this isn't a big part of my research, but there has been work done on controlling Ventonata, most of it kind of out of Idaho and Washington State, and the ARS in Burns has done some good work on which herbicides are most effective for controlling Ventonata. And I don't know how many studies have really tested this at a wide scale, and I don't think much has been done in the scablands, but I think this is a really active area of research. So I think there's still hope for saving these invaded areas. But yeah, I think we you don't have entonata yet, definitely the best, everyone knows the best control is prevention.
Thank you so much, Claire. We're out of time. If you don't mind sharing your contact information in the chat, though, so if folks want to get in touch with you with any additional questions.
Great, thanks, Jen.
Yeah, thank you. All right, so we'll introduce our next speaker is Jonah Piovia-Scott from Washington State University. Jonah, if you want to share your screen and your video.
All right, let's see. It's telling me, I can't start my video. Okay. All right, hopefully that's coming through now.
Yep, that looks great, and you sound good.
Okay, great, thanks. Well, thanks very much for inviting me. I'm gonna change topics, or at least change what kind of organisms I'm talking about, just ever so slightly. So I'm going to be talking about climate change and invasive amphibian pathogens in the Pacific Northwest. So a brief outline of this presentation. So I'm just going to give a little bit of overview on invasive fungal pathogens that threaten amphibian diversity. I'm going to be talking about these chytrid fungal pathogens, of which there are two species, BD and B-Sal. I'll talk about some work we've done to assess B-Sal risk in North American amphibians, and then talk briefly about the potential impacts of climate change on disease associated with these fungal pathogens. So I think it will not come as a surprise to anybody in this audience that invasive pathogens can have devastating impacts on biodiversity. And the impact of these chytrid fungi on amphibians has become sort of a poster child for this phenomenon. So as I mentioned, there's two species. I'm not going to trouble you with the Latin names, but I'll call them BD and B-Sal. And these two species have contributed to the greatest documented loss of biodiversity attributable to pathogens, at least in our sort of recorded history. So as I mentioned, there's two species. They're sort of at two different invasion stages. So BD has a global distribution. Basically, at this point, you can find it just about everywhere we've looked where there are amphibians. It mostly impacts frogs and toads, although it certainly affects some salamander species as well. And it's an ongoing threat to multiple endangered species, including listed species in Western North America, like the Sierra Nevada yellow-legged frog, mountain yellow-legged frog. And in fact, it's one of the primary drivers of the near extinction of those species. So in contrast, B-Sal has emerged more recently, appears to have origins in East Asia, and it actually wasn't described as a species until 2013, when it was found that it was causing these pretty catastrophic population declines in fire salamanders, which you can see in the picture here in Northern Europe. So it appears to have relatively recently spread from Asia to Northern Europe. It's continuing to spread throughout Europe, mostly impacts salamanders. And importantly, it's not known to have invaded North America yet. And one of the reasons that's really important is that North America is a global center for salamander biodiversity. So along with a large group of collaborators, we've been trying to assess the risk of B-Sal to North American amphibians. And one of the main ways we are doing this is by evaluating the susceptibility of different amphibian species to this pathogen. We do that by conducting controlled exposure experiments in the lab. You can see an inside view of an environmental chamber here, with each of those containers has a salamander in it exposed to different doses of B-Sal, this pathogen. To date, we've done more than 50 species, and these were selected based on level of conservation concern. We tried to cover a broad swath of amphibian diversity in North America, so multiple species, multiple families. And we also tried to evaluate potentially important carrier species, so things that are common and therefore likely to play a major role in transmission, and also things that are really good at dispersing and could help spread the pathogen. So a summary of a bunch of these experiments came out earlier this year, and the take-home message is that North American amphibians are broadly susceptible to B-Sal, 74% of, I think this covered 36 species, were infected when we exposed them, and 35% of the species we evaluated experienced disease-related mortality. It was not uniform across different groups of amphibians. So as with previous studies, newts and lungless salamanders were highly susceptible, and lungless salamanders are one of the more diverse groups of amphibians, including in North America, or at least diverse groups of salamanders. Importantly, we also found that frogs and toads can be susceptible and can even succumb to the disease caused by this pathogen. Previous studies had sort of tended to find that, look, this is mostly salamanders, maybe we don't need to worry so much about frogs and toads. But it looks like frogs and toads are susceptible, and that means not only could they succumb to disease, but also they could carry it, and potentially into new areas. So if we take this subset of species that we have looked at using these experiments and make some inferences about, okay, if a species in the same genus was tested and found to be susceptible, let's just assume that that whole genus is susceptible. If we do that kind of expansion, we find that there are possible declines and extinctions in more than 140 amphibian species in North America. The geographic distribution of that risk is not uniform. I'm going to start by showing you sort of some of these components. At the top left there, we have this, what we call an integrated assessment of risk of decline. So that includes information about climate suitability for the pathogen B-cell, host suitability, meaning can the hosts in a particular location get infected? What proportion of them can get infected? And host mortality, what proportion of the hosts in a given location have been shown or can be inferred to experience disease-related mortality. So that's that plot on the top left. On the bottom left, you can see salamander richness. Where are the most salamander species in the country? And when we put those together, we can see that the highest risks of biodiversity loss, potential risks of biodiversity loss associated with B-cell, if it is introduced, are in Appalachia and across parts of the eastern seaboard and on the Pacific coast. So that includes a number of species that are native to the Pacific Northwest. I'm just giving you a rundown of the Pacific Northwest species here. And again, you can see that these two families, newts, which are the Salamandridae, and Plethodontidae, which are the lungless salamanders, that's where most of the susceptible species are. We've assessed a bunch of different families and found that they were not infectable in the lab in our studies. But clearly, there's specific species in this region that we are worried about. So a couple of quick take-home messages. B-cell poses a clear threat to many North American salamanders. Frogs can also be infected. The Pacific Northwest is one of the most at-risk regions. And there's ongoing efforts to develop management strategies and delay introduction. And if you're interested in learning more about those, I encourage you to go to this salamanderfungus.org website, which is the website of the North American B-cell Task Force. And I also want to highlight a new project that I'm involved in, funded by the National Science Foundation, which is looking at how pathogens spread through wildlife trade networks, and how we can foster healthy trade that does not spread pathogens. We think that B-cell got from Asia to Europe through the pet trade, and we think the pet trade is likely the most likely avenue that it could come to North America by. So there's a lot of concern about that. Okay. Let's switch gears a little bit to talk about climate change and B-cell. And I'm going to preface this by saying both of these fungal pathogens tend to not tolerate warmer temperatures. So both B-cell and BD die at temperatures, not too far above room temperature. So this is a projection of current climatic suitability for B-cell in the continental US. You can see, of course, here in the Pacific Northwest, we have some of the most suitable habitats for B-cell, at least from a climatic perspective. And not surprisingly, given the lack of heat tolerance for this species, when you project climate into the future, you see moderate reductions in the suitability of habitat. You can see on the bottom right there, some of those sort of intermediate colors in eastern Washington, Oregon, Northern California, Nevada are fading out. And there's a bit of a decrease in those sort of hotter colors that you see on the coast and in the Cascades. A similar phenomenon may occur for BD. We have a lot more information about environmental associations with BD because we have a lot more information about where BD is distributed. Unfortunately, this information has tended to make the picture more complicated. So while we know from lots of laboratory studies that these pathogens don't tolerate warmer temperatures, figuring out exactly what the environmental drivers are from empirically collected data in the field is not so simple. Although temperature almost always comes out as one of the more important drivers. But one of the reasons it may not be so simple to track relationships between infections observed in the field and environmental variables is that, you know, disease is this sort of emergent property of the environment, the host, and the pathogen. So epidemiologists call this the epidemiological triangle. And I really want to use it as a device here because I think it emphasizes the fact that getting a diseased organism, it can appear simple, but it can also be somewhat complex. So when we evaluate these climate suitabilities, like I was doing on the previous slide, we're just looking at environment. We're saying we think we know something about what makes a suitable habitat for the pathogen, and we're looking at environmental predictors of suitable habitat for the pathogen. But it is also quite likely that impacts of climate change on disease result from environmental impacts on host susceptibility. So I'm going to finish up by talking a little bit about that in a system that I've been studying for a long time. So Cascades frogs are native mostly to the Cascades Range in the Pacific Northwest. They also occur in the Olympic Mountains. And juvenile Cascades frogs in Northern California, where my long-term study sites are highly susceptible to BD, and ongoing population declines and extirpations in that region are likely driven by this pathogen. And so just for those of you that don't think about amphibians all day like I do, when I say juvenile frogs, I mean the tiny guy you see there on the bottom left. It has recently metamorphosed, so it used to be a tadpole. Tadpoles tend to not be very susceptible to BD. So it is, resorbed its tail, grown legs, and crawled out of the water. And once it does that, it becomes highly susceptible to BD. And we think that one of the main drivers of population declines is mortality during that first winter. So they'll come out of their little breeding ponds, they'll get infected with this pathogen. It'll start snowing because these are montane frogs, and then you'll never see them again after that first winter. They just don't come back out of their winter hiding places. One of the things we found recently is that we see more BD in frogs that are these juvenile frogs that are emerging from warmer, drier pools. So this is from a series of meadow breeding habitats in Northern California. On the y-axis, you can see BD prevalence in the recently metamorphosed frogs emerging from those habitats. And on the x-axis, you can see 3 different breeding types that are basically moving towards warmer and drier as you go from left to right. Interestingly, on the far left are springs, and those tend to also feature pretty high BD prevalence. Those aren't ideal breeding habitats because they tend to stay cold, even though they retain water. And the amphibians don't develop as fast in cold water, and it also may be that the pathogen likes it better in the cold water. That middle spot there, that's like great breeding habitat. The water warms up, but the pool doesn't dry up too fast. Whereas as you move over to the right into the pools there, that's where you're starting to get pool drying up as the summer wears on, maybe even starting to dry and making the amphibians metamorphose before they'd like to, and the water is getting quite hot. And of course, as the climate changes, these montane wetlands are among the more susceptible habitats to changing climatic conditions. So the probability that these sort of nice breeding habitat wetlands are going to experience increased drying and accompanying warming temperatures is quite high. This is a sort of older paper, but I think more recent assessments would confirm this pattern. And here I'm just showing the Western Washington here. You can see the Olympics on the left and the Cascades on the right. And yeah, really dramatic changes in the drying probability for these wetlands that have intermediate hydro periods. So to sum everything up as much as I can, BD is an ongoing threat to amphibian biodiversity. BSAL is a potentially catastrophic new threat, and changing climatic conditions may reduce habitat suitability for the pathogens, but they may enhance disease susceptibility for hosts. And finding, what real outcomes are for threatened amphibians given these sort of potentially counteracting effects is one thing we're doing as part of a new project funded by the Northwest Climate Adaptation Science Center. And tons of people and funding sources to acknowledge, and I probably haven't gotten them all on this slide, but I'm happy to take questions if there's time.
Thank you, Jenna. That was really interesting. Yeah, we do have a question for you. This is from Brittany Barker. She's asking, regarding BD, is it possible that the young frogs are more stressed in the warmer, drier ponds, which enhances infection rates?
Yeah, I think the short answer to that question is yes. And it's sort of figuring out the role stress specifically plays versus other physiological processes is something that we're really interested in. And I would say we don't have great answers yet. So to provide a little bit of context, one of the things that happens during metamorphosis is that as the bodies of these animals are completely changing, their immune system more or less shuts down. So frogs do have immune systems, but that period of metamorphosis is a time when the immune system is not very active. And so one of the things that we think is really important here is how quickly does it turn back on after metamorphosis? Because we know that's when they're getting infected. We sort of have data that if you catch them right at metamorphosis, and we do take a skin swab because it's a skin infection, and like right at metamorphosis, the detected prevalence is pretty low. But within a couple of weeks, the detected prevalence just skyrockets to basically, you're going from like 10% to 80, 90% in the span of a couple of weeks. And so we think that has a lot to do with how quickly the immune system sort of reactivates, and that may be linked to stress.
See any additional questions, but we've got a minute or two if anyone wants to throw one in the chat real quick. We do have some kudos though that this was a very useful and informative body of work. So awesome. Yeah. Any additional questions? We'll have here we go. Are there any geographic areas that are at particularly high risk for introduction from the pet trade that you mentioned earlier?
Yeah, so it's a great question. And a lot of the assessments, if you go look at that, the Gray et al. 2023 paper, which I should put a citation somewhere, and some of the other papers that paper cites, one of the things people have tried to do is incorporate that into the risk modeling, and they've done so in different ways. So earlier efforts were like, okay, well, what are the main points of entry, right? And so that was incorporated into some of the geographic risk modeling. In the Gray paper, we sort of had this integrative metric of like, well, it's not just ports of entry. Where are there lots of pet stores, for example? And then in that sort of follow-up paper where we looked at climate risk, we took some different approaches to trying to get at like, okay, well, because we think that sort of spillover event is likely to be somebody that's exposed to an infected animal from the pet trade that then enters a habitat where there's susceptible hosts. And so we tried to do some more interesting things in that Grisnick paper, including like, okay, well, where are people visiting? You know, trying to get metrics of visitation for, you know, national parks and monuments and sort of recreational areas. I think it's a tricky thing. It's all a little, to my mind, it's all a little hand wavy. We're sort of trying to take that into account, but it's sort of, you know, I feel like we're doing the best we can with the way we think it will work. But it's a tricky issue. Oh, I think you're muted, Jen.
Yeah, sorry about that. Thank you very much. We're out of time. Yeah, fabulous body of work. And someone mentioned, could you please post the link to the BSAL risk assessment paper?
Yes.
All right. All right. Thank you so much, Jonah. All right. I would like to introduce our final speaker of the day, Kate Wyman-Grotham. She is with the U.S. Fish and Wildlife Service. Kate, you have the floor. So feel free to share your screen whenever you're ready.
All right, great. Is that
Looks
showing
perfect?
up?
Yep, looks
Awesome.
perfect.
Great. So good afternoon or morning, depending on where you are. I'm Kate Wyman-Grotham. I'm coming to you from St. Paul, Minnesota, where I co-manage the U.S. Fish and Wildlife Service's non-native aquatic species risk assessment program out of our Midwest regional office. I want to start today with a quick review of the relationship between the process of invasion and the options and costs for invasive species management. So we'll assume here that we're talking about a true invasive species, one that spreads and causes harm when it becomes established outside its native range. If we wait to manage an invasion until after the species has established and spread widely, the species will cause a lot of harm and management will be expensive. The earlier we manage in the invasion process, however, the lower the costs and the higher the efficiency of management actions. It's ideal to prevent introductions in the first place, but if an invader slips through our defenses, then we can try to detect its presence as soon as possible and manage while the management costs are still relatively low. One of the prerequisites to successful early detection of an invader is knowing what species we're looking for, so we can use knowledge of the species biology and habitat to search efficiently. That's where horizon scanning comes in. Horizon scanning is a systematic approach to identifying future threats. In contrast to much of the work being done on invasive species, although maybe less so this session, horizon scanning focuses at the margins of current thinking and planning rather than on the introductions we are already actively working to manage. Horizon scanning is foundational to the National Early Detection and Rapid Response Framework depicted in the diagram on the left that the Department of the Interior is currently seeking to implement with input and support from federal and non-federal partners. If we blow up that diagram a bit, you can see that horizon scanning is the very first input to the framework. Horizon scans inform the development of species watch lists that then can be used to identify invasion hotspots to be targeted by surveillance activities. Within the framework, we approach horizon scanning from 3 different angles. Global horizon scans focused on species not yet present within the United States. Asset-based horizon scans focused on protecting valuable assets, and regional horizon scans focused on introduction and secondary spread within the US. The US Geological Survey and university partners are leading several global horizon scans right now for different taxonomic groups, while the Fish and Wildlife Service is leading regional horizon scans, specifically focused on aquatic species. Our objective with each regional horizon scan is to identify species of fresh and brackish water fish, macroinvertebrates, or plants at risk of arriving in a target region of the United States and establishing populations there via unintentional movement. We have completed 5 regional horizon scans since 2019. We have 2 ongoing, and we have 5 more planned. The dominant introduction pathways can vary by region, as can the vulnerabilities of each region to invasive species. So each regional horizon scan is unique in its combination of focal pathways and taxa. So for today's talk, I'm going to focus in on the Pacific Northwest Regional Horizon Scan specifically. And we do have a paper about this that will be published in Northwest Science later this fall. Our regional horizon scans all follow a similar 3-phase process. I'm going to go through the methods and then describe the results for the Pacific Northwest Horizon Scan for each phase in turn. In the first phase, we describe the scope of the horizon scan. At this point, we're not looking for a list of individual species to focus on. We're interested in what pathways and groups of taxa are most concerning. If we went straight to the species level, we might focus too much on species that are already high on our priority list, and we might miss something that isn't on our priority list, but should be. For the Pacific Northwest Regional Horizon Scan, we reached out to a wide variety of partners and stakeholders who are familiar with the region and its invasive species. We sought input from this group via a survey and a pair of webinars on their priorities with respect to pathways, taxa, and the precise geographic bounds of the horizon scan. We had representatives from 24 different entities complete the survey, attend the webinars, or both. These advisory group participants were also invited to provide comments on a draft document defining the scope of the horizon scan based on the information gathered from the survey and webinars. The next 3 slides will cover the 3 dimensions of our chosen scope for this horizon scan. So for geographic scope, we use the term target region to describe the area of concern for species introductions. With input from the scoping discussion participants, we chose the target region boundaries you see here, including the states of Washington, Oregon, Idaho, and the portion of the Columbia River Basin in Montana. Using state boundaries was desired to facilitate a consistent management approach by state agencies within their jurisdictions. At the request of Montana Fish, Wildlife, and Parks, we included the Columbia River Basin portion of that state because of its connection to the rest of the target region and its similar invasive species priorities. The recreational boating pathway was a dominant concern across the scoping discussions, and we selected it as our focal pathway. This pathway is amenable to preventative regulation, but it's challenging for enforcement because it's so widespread across the landscape. Finally, we chose 8 groups of floating or submerged aquatic plants and algae as our focal taxa for the horizon scan. Because these horizon scans are intended to contribute to early detection effectiveness and efficiency, we chose focal taxa based on existing capacity to identify and detect those taxa. The agencies and organizations represented within the advisory group had the greatest existing capacity for early detection of non-native aquatic plants. So taking advantage of this existing capacity means that the project's findings can be applied more quickly and easily. And time is valuable when you're talking about finding an invasive species before it becomes well established. During the second phase of the project, we assembled our initial list of species that could be transported into our target region. We obtained data on the last water body visited by boaters arriving at boat inspection stations in Washington, Oregon, and Idaho. We then used these data to rank other states, provinces, or countries, referred to as donor regions, according to volume of boaters arriving from those jurisdictions. We assumed that the more boats coming in from a particular donor region, the more chances a species from that region has to travel to the target region. The next step in this phase was to look at climate similarity between the highly ranked donor regions and the Pacific Northwest. If the climate in the Pacific Northwest is extremely different from a particular donor region's climate, it's less likely that a species from that donor region would become established. We completed this analysis using a service-developed climate matching tool called the Risk Assessment Mapping Program, or RAMP, which is based on a similar Australian tool called CliMatch. RAMP uses 16 bioclimatic variables and a Euclidean distance algorithm to calculate climate match scores. As the final step in this phase, we used global and national species occurrence databases to identify aquatic plant species in our focal families and genera that were present in those high-ranking climate match donor regions, but absent from our target region. According to the data we obtained from the states of Washington, Oregon, and Idaho, the jurisdictions shown here in blue are the 15 most common locations where recreational boats were last used before arriving in our target region, which is shown in gray. We were able to break down the data by state or province for boaters coming from other parts of the US or from Canada, but we only had data at the country level for Mexico. Most of the top donor regions were classified as having high or medium match to our Pacific Northwest target region. Both of these levels of match are associated with 20% or higher probability of establishment of an introduced species. When we then searched species occurrence databases to build our initial species list, we limited our search to the species present in the jurisdictions with high or medium climate match, leaving out Minnesota, Texas, and Florida. The final phase of the project was to take the list of species present in those high or medium climate match jurisdictions and screen them for potential high risk to the Pacific Northwest. This phase took advantage of the service's established method for conducting rapid risk screening for individual species called the Ecological Risk Screening Summary. Our initial species list from phase 2 included 179 valid species within our focal groups of aquatic plants and algae that are present in those high or medium climate match donor regions from which boaters were most commonly arriving in the Pacific Northwest. We wanted to focus on species that were not already established in the Pacific Northwest, which brought us down to 125 species. Of those, 7 were already present in our risk screening database, so we were able to apply our existing information to the Pacific Northwest target region. That left 118 species. We didn't have the capacity to complete a full risk screen for all of these, but the regional advisory group had advised us to prioritize species that already had a known history of introduction, which narrowed our list down to 27. They also wanted us to prioritize species present in trade pathways, in addition to the recreational boating pathway. Of the 14 species with known trade history, 13 had sufficient information available for us to complete a risk screening report. The other 13 species had no apparent trade history, so we went to the third most important prioritization factor, which is the breadth of the species distribution. We measured this simply by how widespread the species was globally, and 6 species were found on at least 4 continents. We added a final species, starry stonewort, due to interest and information availability, even though at the time it was only found on 3 continents. So as I mentioned before, the service's tool for rapid risk screening is the Ecological Risk Screening Summary, or ERSS. The ERSS classifies risk based on information on past invasive behavior from the literature, climate similarity to the contiguous United States, calculated again with RAMP, and the credibility and reliability of information for performing the screen. The primary output is a categorization of species as high, low, or uncertain risk. But the risk screening report also contains a brief literature review on the biology, ecology, and current distribution of the species, which is especially helpful when a species is classified as uncertain risk. We have an existing library of ERSS reports for approximately 2,000 species, to which the Horizon Scanning projects are now contributing. You can view all finalized ERSS reports at the web address shown at the bottom of this slide. Historically, the standard ERSS methodology only looked at climate match to the current climate. However, we looked at climate match to both current climate and future climate for this project. And we considered 3 climate change scenarios at mid 21st century and late 21st century time steps. We found 5 species classified as high risk to the Pacific Northwest, which means, again, that they have high history of invasiveness somewhere in the world, and they have a medium or high climate match to the Pacific Northwest target region. So suitable climate was found for all 5 species under both current and future climate conditions. These species typically cause impacts by forming dense mats of vegetation with negative impacts to water control infrastructure, property value, and native plant communities. All have either been documented within the recreational boating pathway, or focal pathway, or on other types of aquatic vehicles or equipment, suggesting that they could be also found in the recreational boating pathway. All the remaining species screened were classified as uncertain risk. The 11 species listed on this slide all have a history of introduction outside their native ranges, but we did not have enough information on impact or lack thereof to classify them as anything other than uncertain risk. These species also have medium or high climate match to the Pacific Northwest under both current and future climate scenarios. Finally, these 3 uncertain risk species have a history of establishment outside their native ranges and a low climate match to the Pacific Northwest under current climate conditions. However, that changes to medium or high climate match under at least some future climate scenarios. To wrap up, I'm going to touch on some of the ways in which we can apply these results and the results of future horizon scans in the Pacific Northwest and nationally. Using horizon scans to develop species watch lists helps us focus both prevention and early detection activities on the species most likely to cause harm if introduced. We can include such species as we develop and improve training resources for both prevention and invasive species surveillance activities. We can ensure that we have appropriate detection tools and strategies to be able to detect the high-risk species if they're introduced. Policymakers can consider whether to place restrictions on import or possession of any of the watch list species. Then, especially for those uncertain risk species or for more localized applications, we can conduct further risk analysis. The risk screening conducted as part of our horizon scanning is a very coarse filter for invasion risk. And we can use other tools to get more insight into where an invasive species, an individual species is most likely to establish and what impacts we might expect, even if they haven't been documented yet. Finally, the horizon scanning process we used here is repeatable, so we can return and scan additional pathways and taxa in the future, especially as risks from different pathways change over time. With that, I'd like to acknowledge the folks who provided the watercraft inspection data for this project and the many individuals who participated in the scoping phase of the project. Thank you for your time, and I'd be happy to take any questions if we have time available.
We'll try to do it really, really fast. I think you sort of alluded to this question in your previous slide. Someone's asking, do you know if a similarly designed study is planned to screen the risks posed by invasive aquatic animals, including piscivorous fish in the same region? If not, how might stakeholders initiate a conversation about performing one?
Sorry, my windows are all messed up here. Because I'm still sharing my screen. Okay. Yeah, so our plan, like within the service, is to circle back to each region approximately every 5 to 7 years, so that we can cover additional pathways and taxa. But certainly we'd be happy to work with others who are interested in applying a similar technique to do it sooner than we're able to, since we're trying to cover the whole country.
One more quick question, and I guess that's a good point to provide your contact information in the chat if you don't mind, Kate.
Yeah.
Additional question, how can folks access the other Horizon scans, such as Alaska or the other Horizon scans that are completed?
Yeah, so we don't have final reports for the Great Lakes or Alaska because they were kind of pilot projects to test out different parts of the methodology. But we do have reports available, and I can share the link in the chat for both the Southeast and Pacific Southwest. The two that are currently in progress, those reports will probably come out later this fall or winter.
Great. All right. Thank you, Kate. And thanks to all the speakers in this session. And I think I'm going to pass it over to Emily.